There you go. Thank you so much. Now the floor is all yours.
Thank you for this opportunity. So I'm Moa, assistant professor in environmental system engineering in the Department of Energy and Mineral College of Earth and Mineral Science. So it's my honor to introduce today's speaker, Waleed. So Dr. Waleed Al-Sayeed got his bachelor and master degree in botany and microbiology from Al-Azhar University in Egypt. Also received the doctorate degree in the same institute, where he focused on the developing algal biomass, crop protection strategies, and investigating the effect of some chemicals on the algal genome stability by mapping the distribution of the rhinoclade. Prior joining the Penn State, he was a researcher at the Marine Microbiology Department at the National Institute of Oceanography and Fisheries in Egypt. So he joined our group in the March of the 2022, and here he worked on a lot of work related to the environmental microbiology and the community in the engineered system. So today he will give us the first seminar. So this is small.
Thank you so much. Thank you so much for everyone. Thank you so much for Microbiome Center to including me. Thank you so much for you to having me today. I'm so glad to be here today. Before I start, I need to thank Dr. Jasna. I think she and her group helped me so much in the analysis and training me in the nanobore sequencing, and Tegan, his PhD student, helped me so much in the bioinformatics analysis to understand more about the metagenome and yeah, several things I will explain today. So today I will start. Our topic today is succession of microbial community and antimicrobial resistance in algal-bacterial wastewater treatment system. So antibiotic resistance is a global public health. So and understanding antibiotic resistance is a critical due to the significant impact on environment. The increasing antibiotic in the environment, it's harmful for the environment since the increasing of antibiotic effect on the community and the different kind of microorganisms by transfer the antibiotic resistant gene from antibiotic resistant bacteria to sensitive antibiotic microorganisms. So the environmental antibiotic resistant need for urgent mitigation and improve wastewater treatment planet. It's very, very important to overcome this issue and protect our environment. So in wastewater treatment planet, the sample, the wastewater coming from several categories like houses, building, hospitals, and several industrial sector. During the wastewater treatment, there are a process for biological treatment. During the biological treatment, there are a lot of microorganisms coming from wastewater, including antimicrobial resistant gene. And during this process, maybe the antimicrobial resistant gene transferred to the sensitive microorganisms for antibiotic. So it's very important to take care about the biological treatment during the treatment of wastewater. One of the most promising biological treatment is algal-bacterial system for reducing the pollutant and other unwanted pathogens. So during the photosynthesis, the microalgae produce oxygen, which utilized by the bacteria during the oxidizing of organic matter and transfer it to carbon and other forms that may be utilized by algae or other nitrifier. In the algal-bacterial system, we can reduce the amount of pollutant, and also we can reduce unwanted pathogen. Based on the previous literature, algal microbial community have effect on specific pathogen by increasing the pH and the dissolved oxygen. And the potential of microalgae have some kind of anti-resistant antimicrobial produced in the system. So to quantify the anti-resistant bacteria and anti-resistant gene in wastewater treatment, there are several methods. From microbiological view, the cultivation-based method included membrane filtration method and plate count. It's routine work and can recognize the anti-resistant microbes in the environment and wastewater treatment. But this process is limited due to the interference due to the background of microorganisms. Molecular-based method like qPCR, it's essential and overcome about this issue. But the issue for qPCR, it just recognizes certain kind of anti-resistant gene, not all of gene, and it's very hard to select which kind of anti-resistant gene that we need to monitoring in our system. The metagenomic, it's overcome about this problem since the by doing the metagenomic analysis, we can recognize all of anti-resistant genes that we have in community, and also we can identify the community to understand more about the relationships between and transformation of anti-resistant gene between microorganisms. So I will make a simple comparative between the Illumina sequencing shotgun metagenome and the nanobore, since I'm using this method in my study. So the Illumina sequencing is second generation, but the nanobore, it's third generation. Both of them high throughput technique. Illumina produce short read, and the nanobore produce long read. So the objective of our research to evaluate the succession of microbial community of algal bacterial system for wastewater treatment, and also we try to understand the fate of antimicrobial resistant gene during the wastewater treatment by using algal bacterial community. Moreover, we will make a comparative between the metagenome, shotgun sequencing, and nanobore sequencing to identify the anti-resistant gene and the community in our system. Moreover, we employed the Hi-C to identify the host bacteria and also linked the bacteria with target gene to understand the fate and transportation of anti-resistant gene in the community. We designed our experiment based on photo sequencing batch reactor. Our experiment based on the reactor, the capacity of our reactor around 1 liter. So we introduce real wastewater for our reactor every 24 hours, and the cycle started by feeding around 10 minutes in our system. This is automatically, and then the steering start during the operation. The operation was 12-hour light and 12-hour dark. The hydraulic detention time 1 day. And the settling period allowed for biomass accumulation in the reactor calculated to be around 8.9 days. So in our study, we collect our sample from 3 different stages, from influent and reactor and effluent, to understand the fate of antimicrobial resistant gene in our system. And the sample collected as biomass and sent out to analysis of metagenome. And we did the extraction by the same kits that we used for the metagenome sequencing, and we did the nanobore sequencing. After we collect the data, we start to do the analysis for the microbial community and ARG host by using tracking tool. And we identify the antimicrobial resistant gene by using AMR++, and we validate our result based on some specific gene by using the quantitative PCR. So For the high C library, we send the sample out for the company to do the high C and metagenome from the same sample. They extracted the DNA and treated the DNA with a formaldehyde solution to produce cross-linked DNA. And after that, they use it enzymatic digestion and ligation to prepare the library. And after that, they send the library for Illumina, and they use it they used the NovaSeq Illumina for sequencing the nanopore, the high C data. Once they received the high C data, the bioinformatics team in the company start to do the analysis for the high C data, and they send us the data after the analysis. During the operation of our system, we evaluate the efficiency of removal of pollutant like the chemical oxygen demand. So the figure on the left shows the concentration of COD in the influent and effluent during the operation around 51 days. As you see, the concentration of COD in effluent it's higher than the effluent, and the removal efficiency it's increased gradually to reach it around 85% after, and we calculate the efficiency of removal after 3 SRT around 30 days. Also, to recognize the effect, the operation system, we measure the different concentration, different nutrients, ammonia, nitrate, nitrite, and the effluent and influent. As you see in the figure, it's low concentration of ammonia in the influent in comparative between the ammonia and the influent, and the percentage of removal of ammonia around reached more than 90%. But the conversion of ammonia to nitrate, the nitrate is still high concentration. This is due to we don't use carbon source, since in the real wastewater system they use the carbon source like methane or other kind of carbon source to assimilate the amount of nitrate that present in the system. Based on our analysis, we did the alpha diversity to understand the difference in the community present between the influent and reactor and the effluent. So we find that there are a low diversity in the reactor in comparative between the influent and the effluent, and that's mean it's our reactor it's stable to do a specific function to remove nutrients and probably unwanted pathogen. And we did the Baranova analysis to show the difference between the community inside each sample. And as you see, there are no significance between the effluent and the reactor, since the reactor contribute with some microorganisms for the effluent. But there are a significant difference between the influent and effluent or influent and reactor. So in this figure, we recognize some kind of bacteria as a most relative abundance, like Pseudomonada. This kind of bacteria, it's help our system to remove the nitrate, the nitrogen and phosphorus forms from the system, and this is a high relative abundance in our system. Also, we find some kind of cyanobacteria. This kind of cyanobacteria not present in the influent, but start to present in the seed, the first one here. So you can see this color, the pink color. It's some kind of cyanobacteria and contributed with the effluent, but not present in the seed. This kind of cyanobacteria, it's make like a granulation, form the connection between the different bacteria and algae, and also help to remove the nutrients from wastewater. Based on MR++ analysis, and we recognize some kind some classes of antimicrobial resistant. As you see, we have two classes of antimicrobial resistant. High concentration in the effluent after treatment. That means it's the wastewater treatment may be contributed by increased specific kind of antimicrobial resistant gene class in the system. Also, when you double check the other kind like beta-lactam and phenolic and other glycopeptide, there is low concentration in the as a general, but also the low concentration in the effluent in comparative between the effluent and reactor. That means our system may help to remove a specific kind of antimicrobial resistant gene in the system. Also, the high concentration of, we did the normalization to compare between different kinds of classes. As you see, the high concentration relatively abundance of MR plus antimicrobial resistant gene present in the amino glycoside and multi-resistant, some kind of anti-resistant genes. We did a correlation between.
Do those two, the
Those is
aminoglycosides,
which one?
the aminoglycosides and the MLS, do they have a similar mode of action?
Actually, MLS, there are around 3 different modes of action. But I think aminoglycoside is different. Yeah. Yeah, in this figure, we did the correlation between the high abundance genus, 20 genus that we have in the community, and comparative between the high abundance of genus. As you see, there are some high concentration of gene in specific kind. It's correlated with the specific kind of gene. When I double check some kind of gene here, some kind of genus here, I find they're matching with the influent. That means high concentration of antimicrobial coming with the influent. This figure is coming from the high C data, since we have a lot of data related to the high C, but we're not finished our analysis yet. But from this figure, we can see there are a difference between the influent reactor and reactor. You can see here the resistant gene from plasmid in the influent. It's low in comparative between the resistant gene and the effluent and reactor. That means it's the transformation of anti-resistant gene. It's occurred in the reactor and contributed to the effluent. And you can see the most abundance here for anti-resistant gene. It's based on the genomics. That means it's the antimicrobial resistant gene coming with the community from the influent. We plan to do more analysis to understand the specific host and how can the horizontal gene play important role to transfer the anti-resistant gene from the wastewater to the community in the reactor. As I mentioned before, we plan to do validation for our result. So the left figure, it's the result coming from qPCR. There are a matching between the result coming from qPCR and the result coming from the metagenome. For example, the tetracycline O, it's high concentration in the influent in comparative between the effluent and reactor. This is the data from the metagenome. The same for data in the qPCR here. So the high concentration in the effluent and it's very low concentration in the effluent or reactor. Also, we have the sulfonamide 1. This is high concentration in the reactor and effluent in comparative between the effluent. And when we double check here, we will find the concentration of effluent is low in comparative between the reactor and the effluent. based the data collected from nanopore, also we do the same analysis, and the observation it's approximately matching since we when we collect our sample we don't analysis the same sample for the metagenome and for Illumina and nanopore, just make like matching between some sample. So the alpha diversity here show moderate diversity in the reactor in comparative between the infant and F1. The same analysis in the beta diversity, there are a matching and no significant difference between the reactor and F1, and there are a significant difference between the infant and the reactor and F1. The community here also we recognize the same, the most abundant genus. So there are a similar between the data coming from the nanopore and the metagenome by Illumina. Different concentration, but the same genus. So we make a comparative between some specific sample based on the genus and classes. So as you see here, there are a difference, and it's a lot of difference based on the genus between the Illumina and nanopore. So this is because it's cracking tool is very sensitive for the read length, and there are a difference between the read length in the Illumina and nanopore. But when we did the analysis based on the class, we find relatively matching, and so our plan to do more analysis, and probably we can try another method to analyze the microbial community and make a comparative between the Illumina and nanopore and the Illumina and nanopore. Right now, we do more analysis to uncover between the difference between nanopore and the shotgun metagenome, and the high C. We plan to use the data coming from the high C to combine with the metagenome sequencing to identify and understand more about the transformation of antimicrobial resistant genes. Also, we will do more analysis to understand the microbial community and the relationship between the metabolic pathway in the system. Probably this can help us to understand more about how the microbial community, algal-bacterial community, can remove the pollutant and probably contributed or not contributed on the resistant gene. Not only antimicrobial, but also maybe resistant for the betal or any other kind. So the conclusion of our study until now, so our system have ability to remove around 85% of COD and 95 of inorganic nitrogen. And also there are specific kind of classes that we recognize it. Our system can reduce it, this concentration of these classes, but other classes like aminogenic acids and tetracycline increased during wastewater treatment. The most abundant genus, Pseudomonas, and some kind of cyanobacteria, it's play important role to remove the inorganic compound from wastewater. And also the evaluation of anti-resistant gene in wastewater treatment is important to quantify the potential risk and also protect our environment. Thank you so much for the Penn State Microbiome Center for the fellowship that support this work. And also I want to thank Dr. Ivan since he provide us with more analysis for the metagenome and high sequencing. Thank you.
I think we can take one question before we switch speakers, because we do have a second speaker today. We'd like to start with a trainee, as Emily.
Hi, thanks for a great talk. I'm curious because you have both nanopore and Illumina sequencing. If you had positive and negative controls and were able to tell which one could recapitulate the mock community or which one had less contamination between the two methods. So you had both nanopore and Illumina sequencing. Yeah. And I'm curious if you had controls in this data set, which would let you tell which one had fewer which one had less contamination or which one was able to best recapitulate like a mock community?
Yeah, actually for nanopore we used the standard comparative between so we used several standard for community to understand if their contribution or not. But for the metagenome, I'm not sure if they using standard.
Okay. Okay, thanks.
While they switch, one more question. Steph.
I think it's interesting how you talk about phosphorus and nitrogen being a pollutant while they are major nutrients. So I thought about what is the form of phosphorus that comes in and how it's transformed, and then can you actually maybe use that to concentrate phosphorus for reapplying it to a crop?
Yeah, actually, this is a very good question since we start to do some analysis related to the phosphorus, but we don't have conclusion yet. So
think I can add a little bit on. So the phosphorus, since the wastewater is a major source of phosphorus, so if we use the enhanced phosphorus removal, the sludge, the kind of phosphorus we remove in the system in the form of the sludge, or we can potentially, if there's anaerobic digestion, there's a potential precipitation, like the sulfide precipitation at side stream, since that's a high concentration, usually is beneficial, especially if the wastewater coming from the agricultural wastewater, that will be a good source for recovery. But the liquid phase is kind of still low. The major source is coming from the mainstream. So there's also potential. Currently, our lab is working on the diversion of the pretreatment organic matters to diverge it for energy production. So that could be potentially integrated with some chemical reaction addings, and then so have the both the carbon organic carbon recovery and the like the phosphorus recovery at the same time. So that's a potential like we can potentially look at. So yeah, there are also some interest from the utilities to look at this aspect. Yeah. So it's only from the side stream. There's maybe you can recover around maybe 10 to 20% each. Although the side stream contains about the 20 to 30% of nitrogen, but it's still relatively small compared to the overall flow. So that's some perspective.
Okay.
Yeah.
Right, Emily.
Hey, everybody. I am Emily Davenport. I'm an assistant professor in biology, and I'm really thrilled to be introducing Bob Nichols, who will be giving the second part of the talk today. Bob is an assistant research professor in my lab. I was incredibly fortunate because Bob was the very first hire I ever made in my lab. I started in 2020 as a postdoc. And I was fortunate for a few reasons. So one is that he has been a Penn State lifer. So he did his undergrad here, he did a PhD with Andrew Patterson, and it was keen to stay. And so as a very first lab member, he was essential for me getting set up and going. Whenever I had a question, I would just be like, Bob, how does this work here? And he would walk me through everything. It was amazing. The second thing is because he came from Andrew Patterson's lab, he had all of the necessary experience to just jump in and get started right away. So he does everything in the wet lab, he does everything computationally as well. So he's really a Bob of all trades. And I turn to him for a lot in the lab. And the third thing that is so wonderful about Bob is his attitude. He has the most amazing, like, hell yeah, let's just try it attitude. And so anytime I have kind of a crazy idea or something I know is gonna be a lot of work or a little bit tough, I always lean on Bob to do it because I know he'll just be like, that sounds awesome. Let's try it. Why not? And if it doesn't work or if there are hiccups along the way, he takes it in stride and just keeps going with it. So he was the perfect person to take over the project he's going to tell you about today, because it's something brand new in the lab that we're just getting set up. And obviously, whenever you're setting up something new, there's going to be hiccups along the way. So first of all, thank you, Bob, for all of the work you've done over the years, including on this. And of course, thank you to the Microbiome Center for giving us a fellowship to get Bob trained over in a collaborator's lab. And of course, as a thank you for the Microbiome Center, we have a plaque for you for presenting today. So thanks.
So hi guys, I'm Bob. And yeah, today we're going to talk about utilizing co-culture techniques to investigate the direct and indirect effects of the gut microbiome on colonic cells. So just a little bit about what we do in the Davenport lab. And let's just say that we focus in on microbe-macrobe interactions. So what do I mean when I say that? So a couple questions. First off, what kind of factors determine microbiome composition? That can be a couple different things. There are a couple hypotheses out there that host genetics have a pretty large effect in what our microbiome composition is. We know that our microbiome changes over the course of the year, and we know that our lifestyle can also directly affect what our microbiome composition is as well. So, for example, one of the studies that we're involved with involves people in Nepal. There are a couple of different lifestyles that people will fall into, and we're kind of looking at how that lifestyle variation can kind of change the microbiome composition. The second question that we're looking at is, how does the microbiome affect human health and disease? And we can look at that through a couple of different ways as well. So one can be through GWAS, specifically microbiome GWAS, where we can associate different microbes to different disease traits, different SNPs. We can look at the fitness of the actual host. Another study that we're involved with is looking at how the microbiome can affect fly fitness, which is something very interesting. And the last thing is how the microbiome is involved in complex traits, how these different microbes are kind of interacting with each other, and how they kind of can either predispose people to certain disease and such. So the other thing that we want to look at is kind of zooming into this idea of looking at causal relationships between the mucosal microbiomes and host gene expression. And in the lab, we can do this through a couple of different methods. First, we can look at an ex vivo type model. We are collaborating with Hershey and TGen. Hershey is sending us gut punch outs and gut tissue. TGen is sending us lung tissue. We're kind of looking at how we can use these tissue samples to investigate these microbe host gene interactions. Today, I'm going to be talking to you more about the in vitro aspect, looking at different cell culture techniques and how we can use that to kind of investigate this relationship. We are also working with Jordan Benance. We have an undergrad using one of Jordan's old data sets where he introduced a synthetic gut culture into mice and compared that to conventional mice, germ-free mice, and a couple other mouse models. And the last thing, we have a grad student working on an in silico model where we are looking at MGWAS studies and combining that with a single cell model of a mouse. And we can kind of look to see where these micro-associated SNPs are in the actual mouse tissue, which is pretty cool. But today we are looking at more of this in vitro method. So kind of zooming more in on this question, we're looking at how can we explain these relationships? So there are a couple different ways that we can do this. Like I mentioned before, we can look at these microbiome-associated GWAS studies and get these bacteria that are associated with specific SNPs, specifically in disease states. But the issue with this is it's very difficult to actually confirm these results. With sequencing, we get kind of like a snapshot. We can see that a certain bacteria is associated with diabetes, but it's very hard to actually confirm that. Another thing is that there are a couple hypotheses that show that there is a potential relationship between host gene expression and the microbiome composition. And there's also an opposite relationship where the microbiome composition could maybe affect host gene expression, but we don't know directionality. So how do we actually investigate that? Well, one way is looking at these co-culture techniques. So these can provide a direct look at these microbiome host interactions. Now we can do a traditional co-culture where we have a specific microbiome composition that we can directly expose to the colonic cells. We can take these colonic cells, use CRISPR to knock out genes, and we can look at basically directionality that way by saying, okay, if we knock out this gene, maybe certain microbes will decrease. We can also spike in increasing amounts of different microbes where if we have an association of a specific microbe, we can look say, okay, if there is actually association, the gene expression should change as we spike in or take out a certain microbe. We can also use a transwell, and that's kind of what I'm going to talk about to you guys today, where we can use a transwell to essentially separate the bacteria from the colonic cells. And we can look at direct and indirect relationships of the microbiome. So when I talk about a transwell, this is kind of the thing that I'm looking at here. I don't have a mouse. That's fine. So basically what we're looking at is we have a transwell which sits inside of the media or the cell that we have, the well that we have the cells in. And it essentially blocks the bacteria from contacting the colonic cells. So the transwell is really nice because it has a 0.4 micron filter. So the metabolites from the bacteria can transverse, and also the metabolites from the colonic cells can go interact with the bacteria, but the bacteria themselves cannot contact the colonic cells. And this creates an interesting relationship because if we think about our actual gut itself, we have the mucosa that is basically preventing a lot of these bacteria from contacting our actual colonic cells. Now, there are certain situations where there are direct bacteria cell contact, but for a lot of it, there is kind of a barrier. So this transwell is kind of acting as this barrier. So we can kind of get a better picture of what's going on in the actual gut. So I was lucky enough to collaborate with the Luca lab, Francesco Luca over at Wayne State. And they do a lot of cell culture work over there, and I have done none. So we thought it would be a good idea for them to work with this transwell and for me to get trained, because this was something new for both of us. The plan was to use H-coepic cells and fecal bacteria from several donors that they got from UBI. So the general idea was to set up 4 different treatment scenarios, essentially. We have 4 different categories. The first one is kind of what I talked about before, where we have this indirect treatment where the bacteria are sitting in the transwell. We have the indirect exposure to these metabolites. We'll also have a direct treatment where it's a traditional co-culture experiment with bacteria sitting in on the cells, direct bacteria to cell contact, direct metabolite mixing. We'll also have 2 different types of controls, where we have an indirect control where we introduce the transwell to just the colonic cells. And then again, you'll have the free metabolite transfer, but no bacteria. And then just a basic well with your cells in it, no bacteria, no transfer. So here are the 4 types of things that we're going to be looking at. And we can basically, after the experiment, we'll collect the supernatant to do metabolomics. I was going to do both NMR and LCMS. And the Luca lab would do the RNA-seq on the actual cells. And from that, we can get metabolomics and some gene expression levels as well to compare. And from this, we can look at the direct and indirect effects of the gut microbiome. So I just kind of briefly talk about our plate design. So we had essentially this twice. One plate will have the transwell, one plate will have no transwell. We have 3 treatments from donors, and we would have 4 replicates of each donor treatment, 8 controls per plate, and we would get a nice amount of results to compare and contrast each other. We're really excited for this, but of course, with cell growth, you have issues. So what we wanted was something like this. You can see the cells are nice, compact, ready to be split, really nice confluence, but what we had was something like this, very sad. They were not ready to be split. It wasn't this bad, but it was around like 50% confluent. We were basically stuck. So essentially I was only there for 6 days, and I had to leave. So we kind of just went with what we had and had to redo our plate design. But why did this happen? So this could happen for a couple of different reasons. When they froze down the cells, they could have easily subcultured these cells into a more slower growing cell state. If they tried to seed on a larger flask, they could basically pick out cells that are slow growing, freeze them down. So when we try to thaw them, they're still slow growing, give us these issues. This is also primary cell line. So these cells don't really like to split a lot, and these were already at passage 2 or 3, so they just could have just been at the state where they're growing slow. But the issue, like I said, I was only there for 6 days. This whole experiment itself was a 4-day experiment, and we had a 1-day delay essentially. So we pushed it back as much as we could, and kind of worked with what we had. The person that worked with Shreya, she basically told me that if she was doing this experiment, she would have pushed it back 2, if not 3 days. So that's one of the important lessons that you can learn from doing cell culture work, is that they behave how they want to behave. And if you're actually explaining an experiment, don't play around holidays, don't play around football games, because you'll probably have to push it back a certain amount of time. So the updated culture design, we're going to stick with our 4 that we wanted to do. We're dropping a microbiome treatment, dropping all the replicates, just basically having an N of 1. At this point, this is just to get me trained and get something to look at to see if, will these results actually work? Do I have enough sample to run metabolomics? So at this point, it's just kind of a proof of concept. Since we had extra plate space, we decided to actually look to see if the bacteria were having any interactions with this transwell, because neither of us have worked with this system before. So we essentially had a couple of wells where it was just media and bacteria with and without the transwell. This was basically just put in there to see if there was any interactions between the transwell and the actual bacteria themselves. Here's the updated plate design. You can see it's very sad. We have only 6 wells with cells in them, but we now have our 4 extra bacteria only wells that we're going to look at. So in terms of results, I have the pleasure of having NMR results done. And the only issue with this is that they're all an N of 1. I have no replicates, so I can't really tell you if these are true or not. But I do want to start off with just the bacteria without the cells. So this is interesting because we have the M1 where it doesn't really look like there's an effect, but the M2 where there's this huge blue column, which is a decrease of these metabolites. Now this could happen for a couple different reasons. So my theory is that this was the last well that we did. So it's very likely that we just didn't have any bacteria present or not enough bacteria. And since we don't have any replicates, I can't tell you if this is true or not. But if I had to guess, I would say that there's no interactions between the bacteria and the transwell, mainly just because this is a system that's commonly used. And if there were effects, we probably would have known about it. But this is something I'm going to repeat just to double check and make sure that there aren't any effects from this transwell on the metabolites themselves. So we'll focus more on the actual results from the co-culture study, the co-culture cells. So this heat map is everything. This is direct, indirect, and controls. And the one thing I just want to mention and point out, something that I thought was kind of interesting, is this leucine to oxoisocorporate interaction here. We can see that as leucine increases, 2-oxoisocorporate decreases, and this makes sense because this 2-oxoisocorporate is a metabolite of leucine biosynthesis and degradation. So it makes sense that as one goes up, the other has the opposite effect. So if leucine goes up, 2-oxoisocorporate goes down. So I was curious if this changed if I broke this down based on direct and indirect treatment. So we have two heat maps here. One is just the direct samples, the other is just the indirect samples. And we can see that there actually is a change in this trend. Starting with the indirect samples, we can see that the trend is the same. With the transwell, we have increase of leucine, decrease of this 2-oxoisocorporate. But if we look at the actual direct cells where the bacteria is sitting on the colonic cells, this trend's gone. So we have either a decrease of leucine, a decrease in 2-oxoisocorporate, or an increase of both. And so this looks like to be something that is actually caused from the transwell itself. Could be something where the metabolite likes to stay on the upper edge and doesn't transfer down. But there is some sort of a distance-based association between this metabolite and leucine that isn't there with the actual direct contact. And that could just be because since the bacteria and the cells are right there, they're having a constant exchange of metabolites, and you kind of have this, as one goes up, the other one goes up, and vice versa. So this is something that I want to explore as we redo this experiment, just because this is something that I can't really make any actual claims because, again, I have an N of 1. I don't have any replicates to compare to, or maybe these are just outliers. So the next steps for me is actually doing this experiment. But we also do have LCMS from this that we're running. Thankfully to Nina, she's doing this for me, which is great. And we're going to compare that to the NMR results just to kind of see if we have consistency between the two. Again, this is something that we haven't done. The Luca lab also hasn't done metabolomics on this. The major issue with that is they only have 500 microliters of supernatant, which is not a lot. So we're kind of see if that's enough, if I need to add more. So there will be some benefits from this. The Luca lab is going to still do RNA-seq on these 6 wells with the cells in them, just to look at difference in gene expression. We did tell them, since it's only 6 samples, and we are planning on redoing this ourselves, if they just wanted to send us the RNA extracts, we will do them ourselves. And then, like I said, I want to actually complete this experiment in our own space. We're lucky enough to have a room now that we can set up for our own cell culture work, which is great. And down the line, after we get the gut system to work, we want to do this with a lung cell line as well. And investigate the same type of relationships, but instead of with gut cells, with lung cells. So before I go, I just kind of wanted to go through just some important things to keep in mind for cell culture, if anybody's actually interested in doing this type of work. So I start with this little scheme here on how I was told to basically start a cell line. So what you would do is you get your primary cell lamp at P1, which is passage 1. You would thaw that, grow it up, and then after that first split at P2, it's best to basically take 2 of the 3 splits essentially and freeze them back down. This will act as your backups for whenever you run out of these cells that you're using for your actual experiment. You'll take that third portion, grow it up a couple times. Essentially, you're going to move it up to bigger and bigger flasks. At the end, you'll be at like P5, P6, and from there, you're going to split everything, and basically freeze down amps. And those at the very end is what you're going to use for your actual experiment. And now you can see that the color is different, and that's just basically because as you passage these cells, they're going to have a different behavior from your original cell. So it's important if you actually do this work that you want to keep as many cells or as many vials of the same passage number as you possibly can, because when you try to repeat it, if I were to do this with a cell amp from P5 and P1, I would basically have different results. So it's important to keep as many vials of the cell at the passage that you do your experiment at as you possibly can. And then that's also why we have the frozen down from P2, because when we run out of those, we can basically grow them up and have similar, but not identical cells for that. The other thing, this is a cell talk, so I need to tell you that 70% ethanol is your best friend. If you do this work, make sure to spray everything down with 70% ethanol. It just limits the amount of contamination that you're gonna have. Also, it's important to remember that the cell lines that we use are divergent from actual human cells. A lot of these cell lines are immortalized, our cells aren't. So as we do this work, we're gonna find certain associations. Those may or may not be true. It's just important to see, it's the same thing as doing work in animal models where we have these effects that may or may not translate over. It's still important work. Cell lines are an amazing proxy. It's just something to keep in the back of your mind as you actually do this type of work. And the last thing I want to talk to you is if you guys actually do want to learn cell culture, I was lucky enough to work with the PSU Center of Excellence in Industrial Biotechnology. I took their workshop called 6 Established Methods in Mammalian Cell Culture. So this course is awesome. It's offered twice a year. It's free for graduate students and master students. If you're like a postdoc or a professional or a PI, you can still take it, but it does cost money. It costs about $1,000. But it is a 3-day course, and they go over how to set up suspension and adherent cell lines. They go over how to use a hemocytometer. You freeze down a bunch of cells, and then you fill them back up and see how many cells died. It's an amazing hands-on experience. You work with, you work at the Sartorius Cell Facility in Chanley, and you can email Wendy Oakes. I reached out to her just to make sure I could give her email out. She said, reach out to her if you have interest of taking this course. Like I said, offered twice a year, and they only have 8 spots open. So if you do want to take it, email them sooner rather than later. And like I said, it's amazing. And with that, I'll just take any questions.
So much, Bob, for a reality check on how it is doing. People come in and this is my result, and it's not how we go. So just reminder, things go wrong and we need to learn.
Yeah.
I want to let everybody know that I have flyers with an RSVP QR code on the back of the room for the November 30, because we need a head count for breakfast. So I'm gonna spread them out, look at them as you are leaving the room. And also, first question we like to take from a trainee. Questions, Bob?
Yeah.
Hi, Bob, thanks for the talk. I'm curious how you would interpret the cell line models in an aerobic environment.
Yeah.
And that's going to dramatically affect the microbial composition.
Yeah, that's good. So I know when we grow them, they are at low oxygen. So there is that in the growing space, they're at the low oxygen level, and when we add the bacteria, that's even lower. But yeah, no, that's an amazing question where we are taking them out a lot, and they're just exposed to the air. And that has effect on the actual cell lines and the bacteria themselves, because again, we're using these fecal bacteria that don't want to have air exposure, and that's just something you have to essentially look at. I haven't seen the gene expression results, but I'm assuming that we would see a lot of changes with that compared to like a normal gut RNA experiment. But yeah, that's something to definitely keep in mind. It's a good point to bring up. Yeah.
I was wondering how you select a medium that is supporting some sort of expected metabolism of your culture, cell culture, and also of the bacteria community. And is your medium selecting for certain members of the community?
Yeah. No.
And how long is this exposure of these two?
Yeah, no, 100%. That's a great question. So the media that we use is essentially we would just use the media for the cells and just hope that the bacteria like it as well. Because for us, the cells are more finicky than the bacteria themselves. Like the cells will only grow in a certain type of media. Most of the bacteria will grow in it, but they have done, I haven't done this, but the people we collaborated with have done 6NS before and after, just to check to make sure things are stabilized, everything is growing, and they didn't notice anything that was actively being selected for with this. And we only, it's only a 24-hour exposure, so it's not terribly long. But that is something that when we do this, I will also repeat this just to make sure that there's not lab effects, because I am sure that there is some selection, but I just want to make sure that's not too crazy.
In the back.
Oh, yeah.
Do you think you could use this method with, instead of human cells below, just have two different microbial communities together, or have like say microbes in the well and like some plant roots if it's like a scaled up version? Like, do you think that would work with these types of inserts?
Yeah, 100%. Yeah, I mean, you can use these transwells to separate any two communities, because essentially you're only letting metabolites flow through. So if you had two bacterial communities, you could absolutely change that up. Other people have done this where they even will flip the transwell over and coat the transwell itself with the cells, and essentially have the pores leading directly to the cells to have like a more kind of even closer contact. So there isn't that like space between the two. One of the issues that I saw with this actually was that the transwell has that 0.4 micron filter, but some of the times, I don't know if it's a surface tension thing, but the actual flow through didn't flow as much as we thought. There was some, but there was still a lot of the supernatant that was left in the transwell and not flowing into the actual well itself. So again, that's something that I need to play with in future studies. But when I was doing the research on this, I saw people digging around that would coat the bottom of the transwell with cells, and then you wouldn't have to necessarily worry about that.
Seth, you get the last word.
All right, well, both talks were spectacular from a methodological innovation perspective, because we saw about high C in the first talk and now the transwell is the second talk. This is great for the fellowship program. Thanks for bringing it to our attention from both groups. The transwell assay strikes me as a real opportunity to not only look for exchanges of interactions with the membrane there, but also it's not just metabolites that'll flow, but it will be phages that'll be flowing.
Yeah, 100%.
And we know that actually some human cells can uptake phages. And so some real innovation in your project would be to sequence the human cells and see what phages they've taken up from the bacteria.
100%.
And if you want to talk more about that, I'd be happy.
Yeah, absolutely.
That's
yeah.
super cool.
No,
There's also these like bacteriophage only infect bacteria and only like impact bacteria. Not the case. Like there are immune pathways in the human gut that can interact with phages.
Yeah, there's definitely a lot of avenues that we want to go down once we get this set up and running. It's one of those things. It's like, okay, we're trying not to like bite off too much. It's just kind of like, okay, let's get this working. Let's see if we can actually grow cells and then go from there. But yeah, no, that's definitely something we want to explore, because that's, I feel like it's a very underrepresented
Well, then the
area.
method will last forever for you.
Yeah, 100%.
Yeah, it's great.
We have 1 minute. One last burning question. Actually, we don't have, it's 12. So let's thank our host speakers. Keep coming through in our newsletter.